Resveratrol is a polyphenol that plays a potentially important role in many disorders and has been studied in different diseases. The research on this chemical began with the French paradox, which describes improved cardiovascular outcomes despite a high-fat diet in the French population. Since then, resveratrol has been extensively studied and shown to have antioxidant, anti-inflammatory, anti-proliferative, and anti-angiogenic effects, with those on oxidative stress being possibly the most important and underlying some of the others. However, many signaling pathways are among the molecular targets of resveratrol. In concert, they may be beneficial in many disorders, particularly in diseases where oxidative stress plays an important role. French paradox. The French paradox is a term that was generated in 1992 based on epidemiological data from French people who had a low incidence of coronary heart disease despite the consumption of a diet high in saturated fat. This prompted researchers to think about a possible reason, and scientists propose that moderate wine consumption, almost 57% of the overall alcoholic drink consumption in France, explained this apparent discrepancy, and further suggest that a decrease in platelet aggregation may be the main factor of the effect on coronary heart disease. In 2009, an investigation by the World Health Organization found that the ratio of coronary heart disease-related mortality was found to be 2- to 3-fold lower in France compared to other countries such as the USA, UK, and Sweden. Additionally, a study was carried out in Copenhagen with 6,051 men and 7,234 women aged 30 to 70 years. In this study, low to moderate wine intake was associated with lower mortality from cardiovascular and cerebrovascular diseases. After these observations, a great deal of attention has been paid to the French paradox, and thousands of studies have been performed on different aspects. The French paradox has evolved considerably since 1992. Several studies indicate that heavy wine drinkers may have almost the same mortality rate due to coronary heart disease. Heavy alcohol consumption also increases the prevalence of myocardial infarction, cardiomyopathy, cardiac arrhythmias, hypertension, hemorrhagic shock, and sudden death, confirming that heavy alcohol consumption is harmful to the cardiovascular system. However, the correlation of the drinking patterns with a higher cardiovascular risk apparently depends on the type of drink. Many compounds have been identified as being specifically abundant in red wine, and many of these have a phenolic structure. The main compounds in red wine include flavonols such as myricetin, kaempferol, and the predominant quercetin, the flavin-3-ol monomers catechin and epicatechin, the oligomeric and polymeric flavin-3-ols, or proanthocyanidins, anthocyanins, phenolic acids including gallic acid, caphteric acid, caffeic acid, p-coumaric acid, and the still bean resveratrol. Thus, red wine appears to be the richest source of resveratrol through the skins, seeds, and stems of the grapes that are used. Resveratrol. Resveratrol is a stilbenoid named trans-3,4,5-trihydroxystilbene and consists of 2 aromatic rings that are attached by a methylene bridge. It is a natural phenol and phytoalexin produced naturally by 72 different plant species, especially grapevines, pines, and legumes. It is also present in peanuts, soybeans, and pomegranates in high concentrations. In particular, Botrytis cinerea infection in grapes leads to the exclusive synthesis of resveratrol in the leaf epidermis and grape skins. Since grape skins are not fermented during white wine production, only red wines contain noticeable amounts of resveratrol. This compound was first mentioned by Monsieur Takaoka in 1939 after its isolation from the root of the white hellebore, Veratrum grandiflorum. The name resveratrol was derived from this source since it is a resorcinol derivative from a Veratrum species. Resveratrol is present in cis or trans isoforms, both of which may be glucosylated, and the major trans isomer is the biologically active one. Resveratrol is also produced by chemical and biotechnological synthesis and sold as a nutritional supplement following its derivation from Japanese knotweed, which is the Itadori plant. Resveratrol was identified to be converted to its monohydroxylated form, polyphenol, by the cytochrome P450 enzyme CYP1B1. Polyphenol possesses stronger anti-tumor effects than resveratrol itself, and its hydroxyl group was shown to increase its inhibitory activity on protein kinases and nuclear factor kappa B and its inducer activity on apoptosis. Also, the actions of resveratrol were found to be related to its 3-O-sulfate metabolites. Resveratrol was found to be metabolized mainly to its glucuronide metabolite in the rat small intestine, used as a model. However, in human plasma, resveratrol sulfate was found to be the major metabolite upon oral ingestion of resveratrol. This sulfation is thought to be the main limiting factor for the bioavailability of the compound. Sulfates were also found to be the predominant resveratrol metabolites in CACO2 human colon carcinoma cells, but this formation could be inhibited by resveratrol itself. In order to reduce the fast metabolism of resveratrol, the stability of resveratrol can be improved by liposome encapsulation. Resveratrol bioavailability is believed to be affected by the food matrix. Human subjects were administered pure resveratrol, and high amounts of glucuronide metabolites were detected in the plasma and urine. However, oral doses of grape juice also resulted in glucuronide and sulfate conjugates in the human plasma and urine. Resveratrol is a multi-purpose compound believed to be effective in improving health and preventing or treating chronic diseases. It has been the focus of many animal and human studies. Its biological activities have been shown to depend on its structural determinants, including the number and position of carboxyl groups, intramolecular hydrogen bonding, stereoisomery, and the presence of a double bond. Trans-stilbene compounds, which possess orthodiphenoxyl or paradiphenoxyl functionalities having a 4' hydroxyl group and a double bond, show high chemopreventive activity. Scientists synthesized resveratrol, analog 4' dihydroxy trans-stilbene with 2 hydroxyl groups in the 4 and 4' position to obtain a molecule more active than resveratrol. This analog was more active than resveratrol in the inhibition of secretion of the potent vasoconstrictor peptide, endothelin-1. To increase the activity, several resveratrol analogs have been synthesized. Researchers have investigated the structure-activity relationship by changing the number and the position of hydroxyl groups. Increasing the number of hydroxyl groups on the phenol rings was proven to inhibit the growth of human tumor cells. Hexahydroxystilbene was identified as the most effective analog in various tumor cell lines, with its high free radical scavenging and, therefore, anti-tumor and anti-metastatic activities. Whether the same structure-activity relationships apply to each of the effects of resveratrol remains to be elucidated. The enormous amount of clinical and animal studies suggests that resveratrol may have beneficial effects to improve health, prevent, and or treat chronic diseases in humans. However, despite a large amount of circumstantial and experimental evidence, definitive clinical studies are largely missing. Moreover, explaining the molecular basis of the beneficial results of resveratrol still awaits further detailed and extensive epidemiological studies. Further studies are needed regarding the genetic factors that account for differences in bioavailability and physiological responses to resveratrol between individuals. It should be clarified whether resveratrol can have either synergistic or additive effects in combination with other therapies.